So back in the 1970s, reagents were developed to identify variation in the HLA region, and those were applied to a whole variety of illnesses, and mostly autoimmune diseases. And so we had this rash of discoveries in the 70s and 80s of associations that involved genes pretty much in the MHC, and then this big gap of close to 20 years where the tools for doing genetics were just being developed. There was a lot of enthusiasm and a vast underestimation of the complexity of the problem. And so what we were plagued with until really the last 5 years or so has been very small sample sizes relative to what's required, which tends to be in the many thousands. And then in the last few years, there's been this explosion of discovery of new genes for not only lupus, but a whole variety of autoimmune diseases. And these studies have taken the form of a case control study done on a genome-wide basis, a so-called genome-wide association study, in which large numbers of cases and large numbers of controls are compared across the entire genome with a variety of types of chips, which capture anywhere from 100,000 to 1 million, and now we're moving towards 5 million SNPs per chip, that is per individual. And those can be done relatively rapidly. Our throughput here is somewhere around 500 cases per week of doing 1 million SNPs. And you generate a list of genes that have statistical evidence. But because you're testing so many things, you need to replicate. So there's a big replication phase that goes on for all reliable studies of this type, in which another set of cases and another set of controls are looked at again with a view towards the initial findings from the first study. Now, the spectrum of genetic variation that's involved in complex disorders like lupus is captured here, where there are common variants in the population, seen over on the right of this slide, which contribute a very modest amount to the disease risk. And most of what's been discovered in the last 5 years have been these common variants with very modest genetic effects. There are very few examples of common variants which have a very large effect, and probably the best example is macular degeneration, which was one of the first discoveries that came out of this approach. There are extremely uncommonly rare Mendelian alleles, and we'll mention a few of those. And then there's a whole area in the middle, which we'll touch on, where the allele frequencies are not so high, and maybe the effects are a little higher. And those have been difficult to get at until very recently. Now, so this is an example of a genome screen. This is called a Manhattan plot, for obvious reasons, that captures these skyscrapers of hits, the main one of which is at the HLA region, but there are a number of others here. This is a study that for the typing for which was done here, led by Tim Behrens, now at Genentech. And there are 3 or 4 or 5 definite hits, and then a lot of stuff in this sort of intermediate range of statistical significance that may or may not be true. So to make a long story short, there have been a number of genome-wide screens done by both Tim's groups, John Harley, a number of people in Europe, and there's a list of genes, this is fairly current, but it's changing all the time, of say 20 or 30 genes that have some level of association with lupus, and which meets very stringent levels of statistical significance. And you can divide these into functional groups.